In neuroscience, attention is defined as the active filtering of behaviorally relevant signals and suppression of the remaining as noise. Attention deficit hyperactivity disorder, or ADHD, is a neurodevelopmental disorder characterized by inattentiveness and affects over 300 million people globally. A better understanding of brain circuits that control attention will provide insight in how attention is disrupted in ADHD patients. The basal ganglia is a part of the brain important for action selection and movement. Recently, there's been interest in how the basal ganglia is involved in sensory processing. The striatum is the primary input structure of the basal ganglia and receives input from all over the neocortex. Within the striatum, there are direct and indirect neurons, called spiny projection neurons, and they split into two opposing pathways defined by their downstream projections. The direct pathway is thought to be involved in go, in initiating actions, whereas the indirect pathway is associated with no-go, or exploring other options. So for sensory processing, we think that these pathways may be important for selecting relevant sensory information in the direct pathway and filtering out irrelevant information in the indirect pathway. The auditory striatum is the extreme posterior region of striatum and receives many auditory inputs and have been shown to be important in auditory decision making. In a task shown previously, mice learned to associate high-frequency auditory cues with the right choice and low-frequency cues with left choice. And during a mucimol inhibition experiment that pharmacologically silences auditory striatum, mice are unable to differentiate between auditory stimuli. And mucimol-injected mice select left and right at about 50-50 chance levels. Now, neurexin-1 alpha is a presynaptic adhesion molecule, and copy number variants of neurexin-1 alpha increases risk of ADHD and also autism spectrum disorder. Regarding the cortical inputs to striatum, our lab has previously shown that there are specific deficits to the indirect pathway in neurexin-1 alpha mutants. This can be seen through heterozygous and homozygous knockouts of neurexin-1 alpha and their decreased evoked spiking efficiency in indirect striatum neurons. So given all this previous work, I'm hypothesizing that prefrontal deficits in the indirect pathway of striatum due to loss of neurexin-1 alpha may result in impaired ability to filter out distracting sensory information. And to test this hypothesis, my lab developed a novel treadmill task. To learn more and see our results, please check out my poster. Thank you.